This afternoon. I have sat in many Zangwill seminars over the past 2 decades, so it's a real privilege but also somewhat surreal to be the person standing at the front and giving the talk. So I'm going to be talking today about a topic that's been of interest to my lab for the past 18 years or so, which is reconsolidation-based treatments for mental health disorders. Now, as I'm sure you will see, it's been quite a journey and we are not quite there yet, but I'm hoping to show you that we have made a lot of progress in this aim of developing reconsolidation-based treatments, even though we still have some way to go. So before I get into the meat of the talk, I am going to just give a little bit of context about my lab. And neither Newnham nor Downing, my 2 colleges, have a nice bridge, so I'm afraid I've stolen Queen's to illustrate this. My lab is really interested in 2 topics that may seem like they're quite disconnected. So on the one hand, we are really interested in the basic processes of memory persistence and updating. So what are the molecular mechanisms, neurochemical mechanisms that support memory persistence and updating? But alongside these very fundamental interests, we're also really interested in the development of new treatments for mental health disorders. And so these might seem like 2 quite different fields of research, but actually there is a bridge between those, and one that was argued for by my PhD supervisor, Barry Everett, along with Tony Dickinson and Trevor Robbins, that one key bridging concept here is the formation of maladaptive emotional memories within mental health disorders, which provides a bridge between these 2 very different fields of research. Now, as many of you will know, my lab is a preclinical lab, so the majority of our work is done in rodents. And of course, that means that we rely very strongly on translational and back-translational animal models. So I would say that the rats are really holding up and underpinning this bridge. And this has led to a very strong side interest as to what can and can't be modelled in terms of mental health disorders in animals and how we should use animals to inform our understanding of mental health, which I won't go into today, but if you are interested in those topics, there are a couple of review articles listed here, and I've just guest edited a special issue on animal models for emerging topics in life sciences. So if you're interested, you can refer to those. So that's what I'm not talking about. Let's move to what I am talking about. And I'm going to be talking about this journey that we've gone on in terms of developing reconsolidation into and exploiting that reconsolidation process to try and develop new approaches to treating mental health disorders. And like all long journeys, and it has been quite a long journey, we can essentially think of this in terms of where we've come from, where we are now, and where we are going. And I'll start, of course, with where we've come from. So this will give you a bit of a background in case you're not familiar with memory reconsolidation, but also sort of give a little bit of the history that essentially the first sort of 10 years or so of these reconsolidation-based studies were really focused on proof of principle. So is it possible to disrupt maladaptive memories and to have long-term impacts on behaviour, often in rodent models? So reconsolidation is possibly best understood in terms of contrasting it with the alternative view, which is the consolidation of memories, the more traditional view. And so whereas the traditional view would argue that we have memories that start off in a short-term store and then are either lost or progress into a long-term store for more permanent storage, the reconsolidation view, and this was argued for by Lewis in the late 70s, suggests that we should actually think about memories in terms of their activity state. So we can contrast memories that are in an active state, where they are short-lived, transient, modifiable, with memories in an inactive state, where they're more stable and persistent. So on the face of it, that just sounds like a rebranding of short-term and long-term memory. But actually, there are some key differences between these theories. Because whereas consolidation argues that that transition from short to long-term is unidirectional, according to the reconsolidation view, it's possible to move between these active and inactive states repeatedly throughout the lifetime of the memory. So in principle, it should be possible to take a memory that is well consolidated, old and well established, currently in the inactive state, and under the right conditions to get that memory to reactivate, or more mechanistically to destabilise back into the active state, where it becomes once again modifiable and transient and unstable. Now, we think that reconsolidation has evolved as a memory updating mechanism. So while the memory is in the active state, while it's modifiable, new information could be incorporated into that memory. It could be updated, it could be corrected, and then that memory would subsequently reconsolidate or restabilise back into the inactive state in its updated form. So this view of memory was actually first proposed back in the late 1960s, but it fell out of favour because certainly within neuroscience the emphasis became very much on understanding the molecular mechanisms that underlie memory consolidation, and the emphasis there was all about stability. Now in psychology, of course, we'd known for a long time that memories are actually quite malleable, but it took until 2000 with the work of Karim Nader to really sort of bring these fields back together and to give a demonstration of memory reconsolidation that was based upon a really well-defined psychological process of Pavlovian fear conditioning that had a really good underlying understanding of neurobiology and the neurochemistry underlying that. So that is the process itself. It didn't take long after that first paper from Karim Nader for Joe Ledoux, Karim's postdoctoral advisor, and for other labs to start arguing that this could be a way of disrupting old and well-established memories that have relevance to mental health disorders. So Joe Ledoux argued for it for PTSD, and Barry Everett and I and others have argued for it for addiction. Essentially, the idea is that if you could take an old well-established memory, put it into the active state, and then block that reconsolidation process, it should be possible to trap that memory in the active state. And so if you do that, you are then trapping it in a state where it's unstable and transient. In the strongest version of this theory, what this would suggest is that by doing that reconsolidation blockade, you should erase the memory. I think we as a field have dialed that back a bit. It certainly weakens it. But regardless, you should be able to have a long-term impact of those memories, reducing the impact of those memories on behaviour for a relatively short-term intervention. And so Joe Ledoux had argued for this for PTSD based on their Pavlovian fear work. Barry Everett and I argued for this in terms of drug addiction, as did Jane Taylor. Meryl Kint has also done the same, University of Amsterdam, for PTSD and for phobia. Now, of course, that's making an assumption that maladaptive memories contribute to mental health disorders, and that's an assumption that we should address. So, essentially, the argument goes that there are maladaptive emotional memories that form and contribute to the persistence of certain types of symptoms within specific mental health disorders. And probably the most intuitive one is post-traumatic stress disorder, but it also applies to repetitive disorders like drug addiction. So I'm sure we're all familiar with Pavlovian conditioning, so going back to the roots of psychology and behaviourism. So we're all familiar with this idea that a conditioned stimulus can be associated with a motivationally relevant outcome, an unconditioned stimulus, and generate a conditioned response. And of course, this is highly evolutionarily conserved. There's a very strong selection pressure to be able to do this kind of learning very well. So we can study this in humans, but we can also study it in rodent models where, for example, auditory stimuli paired with a mild aversive foot shock can give rise to species-specific defensive behaviours like conditioned freezing. Now, Keane and colleagues argued back in the mid-80s that actually this could be extended to be thinking about disorders like post-traumatic stress as well, and we can debate the extent to which this model really holds. But their argument was that if you imagine a combat stress example, as an illustrative example here, if you imagine particular conditioned stimuli like the sound of gunfire being associated with the threat of serious injury or death, as it would be on the battlefield, that then leads to on the battlefield an adaptive response of fear. But in PTSD, this memory becomes overly generalised and overly strong, such that when an individual comes home, and now that behavioural response is no longer appropriate, they're still showing the same, but now maladaptive behavioural response. And so this is now a maladaptive memory which is having a long-term impact on behaviour. Now, for PTSD, I think it's a bit more intuitive, but actually it's also the case for drug addiction that these Pavlovian memories and instrumental memories affect the persistence of the disorder. So it was argued by Barry Everett, Tony Dickinson and Trevor Robbins back in 2001 that actually addiction can be thought of as involving the aberrant engagement of both instrumental and Pavlovian learning processes that have evolved so that we can forage and find rewards and, have, reproductive success and, fitness in evolutionary terms. But these become hijacked by drugs of abuse. So although it's undeniable that the acts of instrumental drug seeking and drug taking are instrumental in nature, whether that's ordering a drink in the bar, as we may do after the talk, or a rat pressing a lever to get an infusion of self-administered cocaine in an operant chamber, those are instrumental actions. They start off goal-directed at least, and they may progress to be habitual and compulsive. But these instrumental actions are not occurring in isolation, and they occur in the presence of particular context and discrete conditioned stimuli, these environmental cues, people, places, paraphernalia that become associated with the drug high and actually become incredibly potent precipitators of relapse behaviour. So in the same way that we can make an argument for PTSD having an element of Pavlovian conditioning in there, we can also make the same case for drug addiction, that these drug-associated cues, when they've been paired with the drug high repeatedly, can potentially lead to a subjective state of craving, but also then very potently and powerfully drive drug-seeking behaviour even when an individual is trying to maintain abstinence. And of course, the difficult thing with these cues is that people may not actually be consciously aware that those cues are driving behaviour because Pavlovian learning is implicit. So if we accept that maladaptive memories can contribute to mental health disorders and we marry that up with this reconsolidation finding, this gives the idea that you could take these maladaptive memories, induce them to enter the active state again, even if they're old and well established, and then try to disrupt their reconsolidation and thereby reduce the impact that these memories can have on behaviour in the long term. And the earliest stages of reconsolidation research focused on that proof of principle. And actually, when I joined Barry Everett's lab back in 2004, they had actually already established that in principle it was possible to do that. So Joffley, now at Birmingham, but he was a postdoc in the lab after he was Barry's graduate student, had shown in the early 2000s that targeting a specific immediate early gene known as ZIF268 was able to disrupt the reconsolidation of both fear memories and drug memories. Now, I'm not going to go into huge amounts of molecular detail, but essentially an immediate early gene controls the expression of other genes and proteins that are subsequently required to maintain synaptic plasticity. So by blocking that sort of very early stage of protein synthesis, then Joff was able to disrupt the memory and to prevent both fear memories and drug memories being expressed in the long term. So in principle it was possible, but there's a problem from a translational perspective, because to target immediate early genes, this requires that you have to inject these compounds, these oligodeoxynucleotides, these tiny little bits of DNA, directly into the brain. And of course, therapeutically, that's not going to be feasible. So when I joined Barry's lab, my project essentially was to try and identify mechanisms that lie upstream of ZIF268 that we might actually be able to target systemically with drugs that we could then potentially use in humans. And there are many different neurotransmitter receptors that we could have gone for, but I focused on two, both of which can be linked biochemically to ZIF268 expression. So here we have ZIF268. If you can see that, let's do it. So here we have ZIF268, and here we have the two receptors that I was interested in. So the first type of receptor that I examined was the NMDA subtype of glutamate receptor, which has been really extensively implicated in synaptic plasticity processes for consolidation, also links up to ZIF expression, as you can see. And then the other type of neurotransmitter that was of particular interest was the noradrenergic receptor, specifically beta-2 adrenergic receptors, which we know are really important for the consolidation of emotional memories. And of course, these maladaptive memories are emotional memories, and also potentially can link to ZIF as well. So essentially then the question became, could we use something that was more translational to try and disrupt the impact of these drug-associated memories on behaviour? So I'm not going to go through all of the methods of all of the studies that I'm going to talk about, but I am just going to take a moment to talk through this one, because the logic of this experiment is the same as all of the subsequent ones that follow. So it's worth just taking a moment. Essentially what you're doing in reconsolidation experiments is a single intervention. So you're training the animals on the memory that you want them to have. You want them to have that maladaptive memory so you can try to disrupt it. And then the intervention itself only occurs on a single day with this brief reminder session. So reconsolidation used to be called cue-dependent amnesia back in the 60s. This brief reminder seems to get the memory to go into the active state. And while the memory is in the active state, you administer the compound that you think is going to cause amnesia. So it's a single treatment. You can then subsequently test those animals repeatedly over an extended period of time, but the only time they've had that treatment is that single intervention session. So on the graphs I show, although they're labelled as being in different drug groups, they're not on the drug at the time of test. So one of the first experiments that I did was to look at whether it was possible to block the NMDA receptor and so indirectly modulate ZIF expression and then reduce the impact of cocaine cues on self-administration behaviour in rats. And so this was actually an experiment where we did administer directly into the BLA. So this was a direct intracerebral infusion, very similar to what Joff had done with the oligodeoxynucleotides, but of course you take these things one step at a time. So I used an NMDA receptor antagonist known as DAP5, but essentially it's blocking NMDA receptors. What we found was that this single treatment was able to reduce cocaine-seeking behaviour supported by the drug cue for up to 29 days after that single treatment. So what we have here is the seeking supported by the cue. These are the same animals tested repeatedly and compared to a vehicle control. Now, I should add, we also had a group that only received the drug treatment and no reactivation. That's an important control because if the memory doesn't enter the active state, you shouldn't be able to disrupt it. I'm not showing those data here because the non-reactivated controls never do anything, but we do have those data, so just to reassure you. Not only were we able to disrupt the reconsolidation of that memory and to reduce the impact of those cues on drug-seeking behaviour for an extended period of time, we were actually able to also relate this to ZIF expression by taking a parallel cohort of animals, and instead of testing those animals, we reactivated them under vehicle or AP5, took the brains 90 minutes after that reactivation session and looked for the expression of ZIF268, finding that in the animals that had undergone reactivation and had received that NMDA receptor blocker, those animals showed reduced ZIF expression compared to animals that had received a vehicle infusion. So we were able to show that link to ZIF. Now, at the time that we were doing this work, everyone was saying it was never going to be possible to do anything with NMDA receptor antagonists in humans. So I've looked at the resurgence of ketamine in the mental health literature with a bit of a wry smile. But we were also then interested in looking at this other neurochemical system and looking at adrenergic receptors. So we did essentially the same sort of experiments, but this time using a beta-adrenergic or beta blocker known as propranolol. And so we did this, first of all, for cocaine, but subsequently Moritz Schramm, one of my graduate students, did the same thing for alcohol and essentially saw the same pattern of results. So this time, giving systemic injections of propranolol, we were able to reduce the impact of these drug cues on drug-seeking behaviour for both cocaine and for alcohol. And importantly, Moritz also included a separate control. So he also had a group that received injections of nadolol, which is a beta blocker that doesn't cross the blood-brain barrier. Of course, giving systemic beta blockers, we know that there will be peripheral effects. So by giving nadolol and seeing that there was no amnesia associated with nadolol, that tells us that the effects of propranolol are mediated via the brain, even though we're giving the drug systemically. So that was all very nice, but we were looking in these studies, these first studies, at a very specific type of impact of that drug cue on drug-seeking behaviour. We were looking at a specific process known as conditioned reinforcement and the ability of a drug-associated cue to support new and flexible drug-seeking behaviour. But that's not the only way that drug cues can impact drug-seeking behaviour. So there are at least, if we turn to the learning theory literature, there are at least 3 psychologically and neurobiologically dissociable means by which these drug cues can influence behaviour, which Barry and I in this review termed the 3 routes to relapse because we thought that sounded quite catchy. So we'd looked at this point so far at conditioned reinforcement, the ability of the cue to maintain responding over delays and to support flexible drug-seeking behaviour. But in fact, these cues can also act to support conditioned approach, which if you're approaching the drug cue, you're generally bringing yourself into the vicinity of where you might make that seeking response. And they can also act to support condition motivation, reinstating the relevant motivational state for drug-seeking behaviour or biasing memory retrieval towards drug-seeking. So the question remained in these early days about whether we could also disrupt those other routes to relapse. So we examined this time actually turning to alcohol just for practical reasons, because it's slightly easier to do this with a drug that has a consummatory response. These data you've seen already. These are the same condition reinforcement data that I just showed you. But these other two measures are measures of conditioned approach and condition motivation. And so when we gave, again, the experimental logic is the same. It's just a single drug intervention in combination with reactivation, although the training and the testing is different. Essentially what we saw was that when we gave MK801 and subsequently tested the animals for conditioned approach, they lost the discriminated conditioned approach that they had previously acquired towards an alcohol-associated cue. Now, admittedly, this looks like their CS mind is responding. They're responding to a cue that doesn't predict alcohol has gone up, which is true. But actually, if you compare this to their responding on the first day of training, it's remarkably similar. For condition motivation, we were using a procedure known as Pavlovian instrumental transfer. And the way in which I'm representing these data is that we're comparing responding in the presence of the cue and the absence of the cue. So if their level of responding is the same when the cue is there or when the cue is not, then they will respond at 0.5, so at the dotted line. Anything above that is an enhancement in instrumental seeking behaviour when that cue is presented. And annoyingly on this procedure, it's not quite as clean as I would have liked it. They generalised a lot across the two CSs, but what you can see is that the vehicle-treated animals showed an enhancement in seeking behaviour, whereas actually the MK801 treated animals weren't statistically different from when the cue wasn't on at all. We also looked with propranolol, but there we had quite a different pattern of results. So again, these are the conditioned reinforcement data that I've just showed you, so these are Moritz's. But actually, we found there was no impact of propranolol on the reconsolidation of conditioned approach or condition motivation, at least at the dose that we used. Which was a bit disappointing, but actually then made some of the data that we also had a little bit more interpretable. Because the really nice thing about these learning theory procedures is you can be really, really precise about which psychological process you're disrupting. But obviously you're isolating those psychological processes, so they have limited applicability to the real-life situation. So if we think about one of the rats sort of working for a drug in the Skinner box, actually all three of these associations can be acting simultaneously, especially if we test the animal on the response that he's already learned to make for drug. So if we train an animal to lever press for a drug and a Pavlovian cue, the presentation of that cue and knowing the location of that cue will bring the animal towards that cue through Pavlovian conditioned approach or sign tracking. And that brings them into the vicinity of where they can make the instrumental response. The fact that the cue is present when the animal has access to the instrumental behaviour will be increasing that instrumental behaviour through Pavlovian instrumental transfer, and the fact that they're responding in extinction means that their responding will be maintained by condition reinforcement. So all three of these routes to relapse are acting simultaneously on this version of the task. And actually, when we had run animals on this version of the task, we saw very different patterns of behaviour in the animals that had been treated with MK801 versus those treated with propranolol. So whereas for the MK801 treated animals, there was a reduction in their lever pressing, not to zero, but actually to a level that, although numerically higher, isn't statistically different from not presenting any Pavlovian cues in the test session in a control group. For the propranolol-treated animals, there was no effect whatsoever. Consistent with this idea that disrupting all three of these routes to relapse, which we were able to do with the NMDA receptor blocker in isolation, fits with that reduction of the impact of the Pavlovian cues on this more translational model. Whereas in the case of propranolol, we can only take out one of those and the responding remains intact. So by this point, it was clear that it was possible to disrupt reconsolidation pharmacologically, but actually then another line of research, which I'll mention now and then we'll come back to in a bit, is essentially looked at whether it was also possible to try and exploit reconsolidation and to disrupt it using behavioural approaches. So as well as this pharmacology, would it also be possible to kind of tap into that updating function of reconsolidation and manipulate the memory through behavioural approaches? So that's very abstract. I'll make it a little bit more concrete. So let's imagine a situation where we're looking at reconsolidation of a memory. We'll take fear memory as an example. We've got an animal trained on Pavlovian fear conditioning, and in a typical reconsolidation experiment, what you would do is you reactivate the memory to get it to go into the active state, give your amnestic agent, and then what you see is that in your drug-treated group, as shown here in the pink bars, you see a persistent reduction in the memory because ostensibly this memory has either been erased or it's been weakened. Now, we can contrast that with another approach, which would be extinction learning. So in extinction learning, this also involves re-exposure to the training environment, but this time you're giving really prolonged exposures, repeated exposures of the cue in the absence of the US. And actually, rather than doing anything to the original memory, what you're doing is training up a separate CS no US memory, which competes with the original memory for behavioural expression. So it's a form of inhibitory learning. The issue with extinction is that it's really contextually specific. So if you test the animals or your participants shortly after extinction training, they tend to show the extinction memory as being dominant. But with time, with a change in context, whether that's internal context or external context, then you tend to see the original memory coming back to dominate behaviour and the fear recovers, in this case, as we're dealing with fear memories. Now, Marie Morphy, working in the lab of Joe Ledoux, wondered whether it would be possible to try and combine these two approaches. So she came up with this idea of retrieval extinction, or extinction within the reconsolidation window. And so the idea here is that you take an animal with a well-trained memory, and you reactivate that memory, so you're putting it back into the active state, and then after a short delay, while it still hasn't yet reconsolidated, while the reconsolidation window is open, you then put them through extinction training. And so the prediction would be that if the memory is updating, it should be possible to take this CS US memory and update it such that it now becomes a CS no US memory. You're targeting that original memory, but through a behavioural intervention. And so in principle, what that should do is give you a reduced fear, both short-term and long-term. Now, Marie had done this. She had demonstrated this in rats back in 2009. This was followed up shortly afterwards by a paper from Daniela Schiller in humans in 2010, although there have been some issues around that paper, so I'd recommend reading Tom Becker's views on that. So for fear memories, it certainly seems to be the case that you can try and tap into this behavioural updating. In fact, it also seems to be the case for drug memories as well. So Lin Lu's group, in this phenomenal paper, in which they looked at cocaine self-administration, opiate self-administration in rats, and then moved to an outpatient population of heroin users, they demonstrated that if you take this population of heroin users, who all, as you would expect, have very strong cue-induced craving at the start of the procedure, if you take those individuals, give them a memory reactivation session, this brief reminder of the drug cues, and then put them through prolonged extinction training, after a couple of days treatment, and then assess them for their relapse and their cue-induced responses up to 6 months later, you can see that this extinction within the reconsolidation window markedly reduces the ability of those drug cues to drive craving even 6 months later. So the key group to look at here are the ones in red. These are the individuals who had the reactivation and extinction within the reconsolidation window, and they're compared to a group that had just standard prolonged extinction training, who of course don't show the effect, and a group that had extinction training but outside of the reconsolidation window when the memory would have already finished reconsolidating. So that's really exciting. We took a slightly different approach to retrieval extinction, but we have done some work on that, and I'll come back to that shortly. But there are some similarities with some of the collaborative work that my lab did with Emily Holmes. So, of course, Emily Holmes is really interested in mental imagery, and she's particularly interested in flashbacks and flashback-like phenomena in PTSD. So she has this very nice procedure known as the trauma film procedure. It's very nice theoretically, probably not very nice to do, where you're shown these film clips that are full of disturbing material. So it's things like eye surgery, Scorsese's Big Shave features on the list, road traffic safety adverts, and so on. So really quite disturbing material that in healthy participants will lead to intrusive memories for that material for about a week after they've watched it, and then it sort of dissipates away. Emily had already shown that with these really highly visual memories, if you give visuospatial interference tasks during the consolidation period for those trauma-filled memories, so essentially a dual task procedure taxing these sort of visual stores that will be necessary for the consolidation of those trauma-filled memories to then come back as intrusive memories, that you could disrupt that reconsolidation, you could reduce the number of intrusions. And so what we did in this study was essentially the reconsolidation version of that experiment. So participants were trained up on the trauma film procedure, and they were sent away with a diary to record the number of intrusions that they had. So this is before the intervention. You can see that all of the groups are roughly equal, having 3 to 4 intrusive thoughts of these trauma films in that 24-hour period. What we then did was to essentially visually, spatially interfere with that memory once it had been reactivated using Tetris. So we had groups that were reactivated only, shown in green, Tetris only, reactivation plus Tetris, and then we had a no task control. Now what you can see is that over the week after that single intervention, so playing Tetris for about 20 minutes after having the reactivation of these trauma memories, there was a marked reduction in the frequency of those intrusions over the subsequent week. And even when individuals came back into the lab and we tried to provoke intrusive memories with the intrusion provocation task, there was still a reduction in the group that had undergone the visuospatial interference as compared to the others. So this suggests that alongside the pharmacology, we might also be able to use behavioural interventions to tap into reconsolidation. So that's where we've come from, essentially establishing the proof of principle that it's possible to manipulate these maladaptive memories through both pharmacological and behavioural means. That was really exciting. It generated a lot of hype. And then we come to sort of where we are now. And I would say that we're kind of in that difficult second album period where a lot of the early findings, it's now become clear that actually there's a lot of subtleties and a lot of luck in some of those that have subsequently only become apparent. And really I would summarise where we are now in terms of this challenge of understanding destabilisation. So when and why does it happen, and how can we use that knowledge to try and improve treatment? And I'm going to talk about sort of 2 aspects of this. One is to try and understand those behavioural disruption studies that I just talked about, and then the other is to think about instrumental memories. So I glossed over the fact that memories enter the active state. They destabilise and enter the active state, but of course they don't always do that. It's possible to retrieve memory without it going into the active state. And we know that reconsolidation is subject to certain boundary conditions. So there are conditions that determine whether memory destabilisation occurs or not. Now, it was first established in crabs and then subsequently in rats and in humans. It turns out that one of the big drivers of when a memory is going to update is surprise or prediction error, hence the surprise crabs, rats and humans. We know that the presence of new information is a really good driver of getting that memory to update, which actually, if you think of it as being an updating mechanism, makes complete sense. You need something to be slightly different. You've got something wrong, you update that memory with that new information. It really makes sense. But it's not a monotonic relationship between prediction error and reconsolidation. So actually, in a series of studies that I won't go into detail on now, but were reviewed here, Emiliano Merlot in the lab showed this really sort of nice relationship between reconsolidation and extinction, but also the boundary conditions either side of reconsolidation. So essentially what Emiliano showed was that there are boundary conditions that determine the onset of reconsolidation, but as you go and increase prediction error further, it's not the case that you're more likely to engage reconsolidation. In fact, the opposite. So the reconsolidation window will close before the extinction window opens. And so with really prolonged prediction error and high levels of prediction error, what you do is actually make a new inhibitory extinction memory rather than updating the original. And this is bounded by a period of actually apparent stability that he termed the limbo period, but has also been referred to as the null point. So memory destabilisation can only occur when these specific boundary conditions are overcome. There's this limbo period in between reconsolidation and extinction, and it interacts with memory strength and age, and that's something that we're really interested in, but I won't talk about now. Understanding the mechanisms of destabilisation then allows us, though, to try and understand how these behavioural interventions are having their effects. Because one account of extinction within the reconsolidation window, I'm just presenting this in a slightly different way, but it's the same as before, is that we are updating the original memory. But there is an alternative explanation because, of course, the individuals are going through extinction training. So it's also possible that there is something about that re-exposure session that leads to a facilitated extinction. So that when extinction learning occurs, the extinction memory is better able to compete with the original memory. So it can inhibit the original memory more than it would under normal conditions, and therefore would actually represent a facilitation of extinction rather than a disruption of reconsolidation. So we were really interested to try and understand whether extinction within the reconsolidation window actually does depend on reconsolidation or not. And of course, the key process that will distinguish these two accounts is the destabilisation process, because that should only be required in the condition where you need reconsolidation. So using the fact that we know that violation of expectation should drive that, Melissa Wood in the lab essentially sought to replicate the Monfils experiment, so fear conditioning, Pavlovian fear conditioning in rats, giving them a brief memory reactivation session or not, followed by extinction training and then subsequently testing for recovery of the fear memory. And the first thing to note is that she did in fact replicate the original effect, so giving animals a reminder session prior to extinction training led to less recovery of fear than animals that just received standard extinction training. But if she included a group that received a shocked reactivation, so they're predicting shock, at reactivation they get shock, there is no prediction error. So when she included this RET plus group, what she found was that the effect still persisted. So it seems like you don't actually need prediction error in order to see this reduced recovery of the fear memory with this extinction within the reconsolidation window procedure. Now, we were quite surprised when we got that result, so we thought we would check it. We would check it pharmacologically. And Emma Carhill, when she was a postdoc in the lab, then turned to pharmacological means of blocking destabilisation. Now, by this point, we actually had a fairly good handle on some of those neurochemical mechanisms. So we know that it depends on dopamine receptors. We know that it depends on specific subtypes of NMDA receptors, and that you can also use blockers of L-type voltage-gated calcium channels. Emma used the first two, so she looked at two drugs, one drug targeting dopamine receptors, shearing 23390, and a drug targeting a specific subtype of NMDA receptor known as ifemprodil. Essentially, her setup was the same, except that she was also giving infusions directly into the amygdala prior to the reactivation session in an attempt to block the destabilisation of the memory, and then seeing whether this extinction within the reconsolidation window effect would still persist. And her data were a little messy, but if anything, gave us more cause to think that it was a facilitation of extinction, because actually she didn't see the effect in her vehicle group, but she did in the animals that received the sharing 23390, who were meant to be having destabilisation blocked. So we checked again with ifemprodil, and the same pattern of results emerged. So pharmacological blockade of destabilisation didn't prevent this effect from happening, and in fact revealed it when it wasn't seen in the vehicle-treated animals. Interestingly, we then turned to addiction. So I talked through this tour de force of a paper a few slides back, and when Tony Farragher came to the lab to do his postdoc, he wanted to know whether he could replicate this effect in the cocaine self-administration experiment from this paper. So essentially, what he did was to train animals on cocaine self-admin and then put them through this retrieval extinction procedure in exactly the same way as the Lin Lu lab had done previously, again with groups with extinction within the reconsolidation window, so a 10-minute gap, outside of the reconsolidation window, so a 6-hour gap, or standard extinction. And then he tested for the ability of the drug memory to come back under conditions of drug-primed reinstatement and spontaneous recovery. Now, we were absolutely staggered, actually, to see that we did replicate the effect. So cocaine-induced reinstatement was reduced. Spontaneous recovery of drug seeking was also reduced by doing this extinction within the reconsolidation window manipulation. But we were still concerned, especially in light of the fear data, that this might not actually depend upon reconsolidation mechanisms. So we first wanted to establish whether it would be possible to enhance, to produce that same behaviour, but using a procedure that we knew wouldn't engage reconsolidation. So we actually turned to the behavioural tagging literature, which suggests that an arousing and novel experience prior to learning will then facilitate subsequent learning. So we use novel context exposure. So shouldn't reactivate the original drug memory. The animals are nowhere near a Skinner box. They're just going into an open field with different combinations of wallpapers and scents and so on to make it a novel context prior to extinction training. So essentially, Tony replicated or repeated the experiment, but including this novel context group, with one slight change as well. We knew we were going to go on and do some pharmacology, so we also included a second group of extinction within the reconsolidation window, but with a slightly longer delay to give us some more time for drugs to wash out in subsequent experiments. And what we found there was essentially the same thing, that the extinction within the reconsolidation window effect replicated, but we could also see the same effect by exposing animals to a novel context, suggesting that actually facilitating extinction was enough to reduce this drug-seeking behaviour. So then, to try and tease apart sort of conclusively whether you need destabilisation or not, we blocked destabilisation pharmacologically, this time using an L-type voltage-gated calcium channel blocker called nimodipine. And for completeness, we also did this for the novel context group as well. Essentially the same experiment, this time involving injections prior to the reactivation session. And as you can see, these two graphs, so here we have the vehicle data, here we have the nimodipine data, and these graphs basically look the same. So pharmacological blockade with dopaminergic receptor antagonism, glu-N2B selective, NMDA receptor antagonism, L-type voltage-gated calcium channel blockade, all of these different neurochemical mechanisms underlying destabilisation, the effect persists, which makes us think that even across these fear and drug memories, that actually these behavioural procedures may in fact be facilitating extinction rather than actually engaging reconsolidation mechanisms, which is not necessarily a bad thing, but it's worth knowing those mechanisms because the way that you would try to optimise those therapies will be quite different depending on whether you're targeting reconsolidation or extinction. So moving on to sort of the next part where destabilisation is an interesting challenge, I very proudly showed you sort of a few slides back these data where I said that we were able to reduce seeking behaviour down to the level of omitting the CS in this sort of more translationally relevant relapse procedure. But one thing that is clear from these data, and that I glossed over, is the fact that the animals also have an instrumental memory for which lever used to be paired with the drug. And you can clearly see that they know which lever used to be paired with the drug. They don't need the Pavlovian cue to support that. There's a separate instrumental memory that clearly is not impacted by this manipulation because all of the animals still discriminate between the lever that used to give the drug and the lever that didn't. And instrumental memory reconsolidation has been quite a controversial topic. So there was a paper fairly early on, 2004, suggesting that instrumental memories didn't reconsolidate. That kind of shut down the field for about 10 years, but essentially there are some issues with that paper, particularly the fact that action outcome and stimulus response associations can to some extent compensate for each other. So we decided to revisit this, actually around the same time that Geoff Lee did as well. He was slightly more successful than we were. So the first time that we decided to try and address this, we thought that we would apply the same kind of ideas that we'd applied to Pavlovian learning. So I showed you a bigger version of this diagram a few slides back. Essentially this idea that if you give sort of repeated prediction error, we know that at some point we're going to engage extinction. This is true for instrumental extinction as well. So actually there should be a point, if we make the reactivation session short enough, there should be a point where we can target the reconsolidation window. So we trained animals to respond for sucrose pellets, very standard, sort of FR1, press a lever, get a sucrose pellet reinforcement schedule, and then just gave them varying lengths of time in which they were responding in the absence of sucrose reinforcement. However, no matter how short we went down to 1 minute's reactivation, we just didn't seem to be able to get any effect of MK801 to induce amnesia. So we clearly saw a blockade of extinction learning when we gave these long re-exposures, but we seem to be hitting that limbo period even very, very early in that re-exposure session. Now, shortly after we collected these data, Geoff Lee's lab published data suggesting that actually non-reinforced reactivation isn't effective for instrumental memories. And thinking about it, it kind of makes sense. And the reason that I say that is that I've talked a lot about prediction error as being important for driving memory updating, but what I haven't talked about is priors, because that expectation has got to come from somewhere. And reconsolidation as a field has focused very heavily on prediction error, but not very much on what that expectation is in the first place, and that's going to make a difference. So this is not a model, this is me drawing Gaussian curves. I'm talking to people in engineering to make this a proper model, but at the moment this is just sort of theoretical. But essentially we can think of when are we going to drive memory updating? It's going to be when the sensory input fits within our current expectations. So if the sensory input is very clear and really different from what's gone before, as might be the case in those non-reinforced sessions for instrumental memories, there won't be any memory updating because it's clearly a different situation and a new memory will be made instead. If, however, we have a much more imprecise prior, then maybe that same precise sensory input overlaps enough that it will drive memory updating. Ideally, we'd have like a nice balance where we've got a little bit of imprecision, but enough to sort of drive updating. And possibly, in theory, if we had really imprecise sensory input, maybe that's not reliable enough that you would update your memory because the weight would just be too volatile. So we then started to turn to that kind of thinking about how we might disrupt these instrumental memories, building on what had been done before by Geoff Lee's lab. And so Geoff had shown, first of all, non-reinforced sessions don't lead, reactivation sessions don't lead to instrumental memory destabilisation. We'd seen that already, we replicated that here. But what Geoff showed that was really interesting was if you take animals that have been trained on one very predictable schedule, so a fixed ratio schedule, you respond this many times, you get a pellet, and you move them onto another very predictable schedule, you also don't see memory destabilisation. So if you take animals that are trained on FR1, then put them onto an FR20, you don't see any effect of MK801. However, if you move them onto a more unpredictable schedule, so a variable ratio schedule, then Geoff found that you do, in fact, see a reduction in the strength of the memory. So here we think what's happening is that these are clearly very different situations, so that's not driving memory updating. In the case, though, of the variable ratio schedule, where, in fact, you do see a reduction, not a massive effect, but you do see a reduction, what we think is instead happening is that there is some imprecision in that sensory input. There's volatility in that schedule that then might be enough to allow that to overlap enough to lead to the updating of the original memory. But it's within limits, because if you put the animals onto a completely random schedule, so again the average reinforcement is the same, but this time it's a random ratio schedule, there we don't see a disruption of the instrumental memory. So there we would suggest that the input is so imprecise and so random that that would not drive memory updating. Now this is ongoing work, we're still sort of working on some of the experiments around this, but one that I will present is some of the molecular correlates. So Harry Robson, when he was in the lab as an MPhil student last year, was looking at molecular correlates of instrumental memory reconsolidation, basically comparing that FR20 condition and the VR20 condition where we do and don't see memory destabilisation. Again, I won't go into all the molecular mechanisms, but what we do know is that when memory is destabilised, there is a molecular correlate in that one specific postsynaptic density protein gets degraded. So this is a protein called SHANK. In the animals that had undergone this VR20 reactivation, so we're comparing them here, so setting the FR20s at 100%, what we found was that there was a reduction of SHANK expression within one region of the striatum, the anterior dorsolateral striatum, in those animals that had undergone that VR20, suggesting that where the memory is destabilising, there may in fact be a molecular correlate in that part of the striatum, which might then support the reconsolidation of that memory. So still correlative, it's ongoing work, that's kind of where we are at the moment. So in terms of, how would I summarise all of that? I I think inducing memory destabilisation is clearly a challenge, but I would argue it's practical, not a theoretical one. So the idea that instrumental memories don't reconsolidate at all, or there are specific types of memory that don't reconsolidate, I'm not completely convinced about, but I think there are practical issues that actually get them to reconsolidate. We just need to be creative in how we do that. So that's where we are now, difficult second album. Where are we going? Now, there's a number of directions that I could talk about here. Certainly some of the work is now getting picked up by people doing human experiments. There are clinical trials. In fact, Meryl Kent has set up a clinic for using reconsolidation-based approaches for phobia and PTSD. I'm not going to talk about that. I'm actually going to talk about trying to apply this approach to other mental health disorders. And the one that's been of particular interest to my lab at the moment is obsessive-compulsive disorder. So obsessive-compulsive disorder, like addiction, is, of course, a chronic and compulsive disorder. It's in a spectrum of compulsive disorders. Really interesting finding with OCD is that patients will sometimes report an alleviation of these obsessive and compulsive symptoms with a change in environment, suggesting that these contexts, these cues, might also be influencing the propensity to show these maladaptive behaviours. So we're really interested in that. There's still a lot of work to be done, I think, to understand the contribution of maladaptive memories, as I'll talk about. But this suggests that there might be a way of trying to bring these approaches to the treatment of OCD. The question then becomes, how on earth do you model OCD in a rat? And luckily, Dawn Eagle and Sharon Moraine Zamir, who's in the audience, had already thought about this problem by the time I came to it. They developed a highly translational, in fact, deliberately designed to be translational, both rats and humans, task that assesses dysfunctional, compulsive-like checking in rats and humans. I'm going to talk through the rat version. Essentially what we have are rats that are responding for sucrose pellets in a really unpredictable situation. So they're responding on a lever that gives them pellets in a slightly, in quite an unpredictable way. So they have to press between 10 and 20 times to get that pellet. They don't know how many times they're going to have to press within that range when they're responding. To make the task even harder, we switch which lever is correct at any point throughout the session. So it's really unpredictable as to what the rat needs to do. But we do help him out because we give him a lever at the back of the chamber that we deem the observing lever. And so when he doesn't know what to do here, he can go and press this lever, and that will switch on a light above the lever that's currently being reinforced. So what he should then do, if he's doing this in an adaptive way, is to go and work on that lever and get reinforcement. When the reinforcement rate drops below a certain degree, then he should go back and check again and check if the contingency is switched. Most of the rats do that, but not all of them. So there are a subpopulation of animals who show this really dysfunctional behaviour, where instead of turning around and going and pressing the lever that actually gives them the sugar pellet, they spend a lot of time pressing this observing lever, which is on the back of the box. It's about as far away as you can get from the pellet and, of course, isn't linked to pellet reinforcement. They're not extending the length of the cue. They're not responding necessarily for the cue because the cue will go off in exactly the same time. These extra observing lever presses have no function and no point to them, and yet some of the animals spend a huge amount of time engaging in them. And so it's that dysfunctional checking behaviour that we're really interested in as being something that is potentially like checking in OCD, a sort of exaggeration of this normal adaptive behaviour to a point where it becomes maladaptive. And as I mentioned, this was designed deliberately as a highly translational task. So Sharon has, running at Anglia Ruskin University, the human version of the task, which has the same fundamental task logic, although obviously the humans get the task a little bit quicker than the rats do. Now, we're really interested in the impact of Pavlovian learning on this task for a number of reasons. And we know, for instance, that individual differences in reactivity to Pavlovian cues seem to correlate with compulsive-like behaviour. So a lot of this evidence has come from the addiction literature, where work on how animals respond to Pavlovian cues, so this sort of Pavlovian conditioned approach procedure. There's a lot of individual variation in what animals do in that kind of procedure. Some animals spend a lot of time engaged with the cue, and they don't spend so much time engaged with the goal location. These animals are deemed as sign trackers. Other animals are the goal trackers. So they see the cue that is associated with the reward and they go to the reward location. And there's been a huge amount of work on these different subtypes of animals, particularly from the addiction field. But there seems to be evidence that sign trackers are more likely to engage in habitual behaviour. There's an argument as to whether they might also be more likely to engage in compulsive behaviour as compared to these goal trackers, who seem to be using more model-based learning. We know from the addiction field that sign trackers will continue seeking drug when it's not available, that they move more quickly to compulsive drug seeking, they're more sensitive to cue-induced relapse. And something that really caught, I think, both mine and Sharon's attention was a finding from Mike Lapelli's lab by Lucy Albertella, where they had essentially a version of something akin to sign tracking in humans, where you're training people on cues that are associated with rewards, of standard Pavlovian conditioning, but then getting them to respond on a target detection task, with some of those distracting cues being the cues that were paired with reward previously. And there's a lot of variation in how distractible people are with those cues. So this value-modulated attentional capture task is similar to sign tracking. It's sort of measuring the same sort of thing, and that correlates really nicely with self-report measures of compulsive-like behaviour, both for OCD-like symptoms but also for addiction symptoms. Now, in work that I did with Dawn Eagle and George Valston, we showed actually across a number of cohorts, in fact we've replicated this at least 3 more times since these data were published in the lab at the moment, that these sign tracker rats, if you classify these animals, these sign tracker rats show really high levels of dysfunctional checking. It's that population who are the ones who check loads. So if you make the task more uncertain than it is already, so here we have functional checking, here we have dysfunctional checking, uncertainty increases checking in everybody, but it increases dysfunctional checking more in these sign tracking animals. If we put the rats into anxiogenic conditions, so if we train them on contextual fear conditioning and then port those cues into the chamber, so we're making them more anxious, then actually, there's a little bit of an effect on functional checking in the goal trackers, but the main effect on the dysfunctional checking is that sign trackers are higher. And then if we actually use real threat, so if we punish when they make the wrong response, when they get the incorrect lever, that increases checking across the board, but dysfunctional checking remains higher in the sign trackers. And interestingly, when we take away the shock contingency in the last 6 sessions here, so these are put into groups of 2, you can see that that increase seen in goal trackers rapidly goes back to baseline, but not the sign trackers, who seem to be more rigid in their behaviour. So what this might mean is that memories, maladaptive emotional memories, could potentially be playing a role in OCD as well, whether that's aversive memories driving functional checking or potentially appetitive memories, which may be driving more kind of habitual instrumental behaviour. So we know functional checking is increased by aversive memories. We know that dysfunctional checking is associated with the sign tracking. And so what we're doing right now is trying to look at whether, how the basic associative structure of this task in a number of different ways, in fact. So one thing we're really interested to know is there something magic about sign tracking, or is this something about an individual difference in the way in which animals respond to Pavlovian cues, the control that those cues have over behaviour? And so another way of looking at that would be to look at a different one of these potential routes to relapse, these interactions of Pavlovian and instrumental cues. And that's work that's being headed up by Felipe Espinelli-Amarin, postdoc in the lab. He's working with the rats. Shira Berg, graduate student in the lab. Emily Breeze is a postdoc with Sharon Moraine-Smear at ARU, where we are trying to establish in both rats and humans the degree to which the sign tracking phenotype correlates with performance on Pavlovian instrumental transfer, and how that also correlates with not only self-report measures in the humans, but also performance on the ORT, a more objective version of those Albertella data earlier. But we also want to know whether sign trackers are more likely to show compulsive dysfunctional checking. This is something that is being looked at by Louise Pickenham in the group, graduate student, We want to try and examine the causal relationship between that. That's something that's being examined by Alice Hong. And ultimately, so where I would like this to go is to really try and understand whether we could actually target the reconsolidation of Pavlovian instrumental memories in a very different disorder, in OCD, at least in our, starting off with our rodent model. So essentially then, where are we going? Well, there are a number of ways that this is going at the moment. Clinical work is ongoing. There's still work that needs to be done in terms of understanding how we can reliably induce destabilisation of memories. But ultimately, where I would like this to go is sort of working out whether we can also use this reconsolidation blockade approach for other mental health disorders like OCD. So I will finish there by acknowledging all of the many people who have contributed to this research, the current lab, the funders, and you for your attention. Thank you very much.
Many thanks indeed, Amy. Our tradition is that we have a little break now where those who have to go away can slip out, and then we have questions. That was lovely, very fluent.
Thank you.
Yes.
Should I leave it recording for questions, or do I switch it off?
Too many questions. I'm going to pick up a small person and then
Okay. Oh, okay. Yeah.
Keep it recording for a while,
Okay.
and because you know your way around all these people, you know how much, I'll let you control the question.
Okay. All right. I'll do.
Until, about, say, 10 to 6.
Okay.
See how it's going. I suspect that Amy knows more of you than I do by name, so I'm going to let Amy control the questions.
Okay, any questions? Colin.
The synaptic tagging capture hypothesis by Richard Morrison, right?
Yeah, so actually that's a really nice question. So I think the behavioural tagging works really nicely with that. So the idea essentially that, when the neurons are undergoing plasticity, that these plasticity-related proteins are getting expressed by a neuron that's undergone recent learning. Then if you have a second event coming in and inducing another, more synaptic plasticity, and another synapse on that neuron, that can hijack some of these plasticity proteins and then strengthen that second learning by virtue of having had the first. So I think behavioural tagging kind of developed directly out of that, and I think Julieta Frey is actually on both of those papers. So potentially, I think, we haven't directly examined it. Obviously easier to do in invertebrates than invertebrates, but I think that certainly for the extinction within the reconsolidation window work, certainly in our hands where it really does seem to be a facilitation of extinction, that something like synaptic tagging, behavioural tagging may well be playing into that and giving us that increase in learning, which is then leading to reduced recovery of the memory because the extinction memory is stronger. Mike?
I'm wondering if you could clarify mechanistically why you think that extinction learning would be enhanced by either a brief re-exposure or by behavioral tagging.
Yeah, so I mean, the idea is essentially that this is an arousing event. And so that may well have, impacts on adrenergic signaling, dopaminergic signaling. There's a whole host of things that could happen neurochemically that would then allow that second learning opportunity to occur more readily and enhance plasticity there. So I don't have any strong, I'm not going to say it's a specific region of the prefrontal cortex because I don't know. But certainly I think that kind of arousal argument, and bless him, Tony had to do this over 14 days and managed to generate 14 novel contexts in an open field, which was no small feat. But it is a novel and arousing experience. So I think the fact that you can still see it, even when this is in a completely different room, there is no similarity with that training context. It just does make me wonder how much of the reactivation is actually more about a generalised phenomenon like arousal and sort of non-specific effects on the learning, which, as I said, doesn't necessarily negate this as a useful tool for treatment, but it means something very different. And then, of course, in terms of optimization, you wouldn't be looking for ways of kind of enhancing destabilization. You're looking for ways of actually facilitating that learning even further, so decycloserine or something like that. So I think the mechanism is important. Clinically, it may well not matter, but in terms of sort of trying to optimize that treatment, I think understanding the mechanism is really important.
Thank you.
Yeah.
Yeah, right. So really on that, you used basically NMDA receptor blockers and beta blockers.
Mm-hmm.
So these are very two broad spectrum targets in the central nervous system. So I wonder if, for example, targeting mechanisms can be utilised to focus on these receptors in specific parts of the brain.
I mean, we can certainly administer drugs directly to the brain. We've done that in some of the studies. There's always been that tension, I think, between the fundamental side of the research, where you want that specificity, and the translational side of the research, where you want to be able to give the same treatment in humans. There may well be some mechanisms by which you can get more specific targeting. We're currently talking to Paul Miller in pharmacology, who's got some really exciting antibodies for GABA receptors. He's working on getting them to cross the blood-brain barrier in rats. And if that came off, that would be a systemic treatment that you could engineer to target GABA receptors in specific brain regions, which would be phenomenal if that could work. But that's still at the level of we are talking about that. That would be a wonderful thing to have, but we're not there yet. So in terms of that, the selectivity, I mean, certainly the selectivity, for the reconsolidation work, part of the selectivity is given by the use of the reactivation session. So there's plenty of work in the reconsolidation literature showing that where you have networks of memories, the ones that are, like if you're reactivating in such a way that you're sort of leaving one of those networks alone, that stays intact. But if you're taking out shared nodes of a memory, if you want to think about it in those terms, that will then affect multiple memories. So actually the corollary of that is that if you really want to get rid of all of like drug-associated memories, you'd reactivate by giving re-exposure to the drug. But of course that then has ethical issues associated with it. I think a lot of that specificity is coming, in fact, from the behavioural reactivation session, so that you can then give these sort of broader spectrum pharmacological treatments that should only be affecting the neurons that are involved in that memory that are currently in this unstable state. Andrea.
So I was wondering whether you can maybe explain or elaborate a little bit more at what level of memory representation you think reactivation and reconsolidation can operate. Because if you think about sort of like PTSD, that might be like one short sort of like really important event that can be targeted. But I think later you alluded to that actually having priors, sort of representation of priors, might also be changed by that. And they might be sort of like a different type of sort of memory and the way you'd be operating it. Whether you think that, like, how that fits together and whether different mechanisms are involved.
Yeah, that's a really good question. So I think actually, how best to address that? So yes, I agree with you, and I think that the kind of models that put this really strong emphasis on Pavlovian fear conditioning and PTSD, of course, are not the be-all and end-all of PTSD. There's clearly much more going on. Even, in fact, in some of the rodent models, it's clear that actually the better rodent models are involving a sensitisation of the stress system that is separate from that Pavlovian fear conditioning process. And I guess it would also depend on when you can intervene, because I agree with you, if that sort of trauma, if that trauma is old and well established and is already being incorporated into, schemas, semantic memory, the way that people interact with the world after trauma is not necessarily how they interact with the world before trauma. There's actually a whole set of representations there that may well not be addressable best through these kinds of therapies, but might be more responsive to something like cognitive therapy. So I don't mean to imply that this would be some kind of magic bullet that would just clear everything. I think where these kind of treatments are particularly useful, though, is where you have these triggers that are not necessarily... There's not necessarily a conscious awareness, or you don't know that you're about to encounter them. And reducing the impact of those on behaviour, I think, would be useful. But I think it would need to be alongside other forms of therapy that are already being used successfully with patients. So this would be an extra thing to add to the arsenal. Any more questions? Yeah?
I noticed that in the example of using a visual experience in Tetris to block the visually encoded intrusive memories of the trauma scene, in that treatment you use the same modality to disrupt the association as you did before there. Trying to now sort of apply that elsewhere. For example, in maybe say in trying to disrupt a smoking addiction, one of the cues is, I'd imagine, the smell of cigarettes. Could you say, as a cue, try to use an olfactory cue like the smell of cigarettes in reactivation, and then during the disruption and extinction, try to do some sort of olfactory interference if that exists?
So I think that's a really interesting idea, because I think you're right that the kind of these dual task procedures are relying on the fact that it is the same modality. I'm sure I have read at some point, but I've never been able to rediscover this paper, where someone looked at the same kind of approach for earworms. You know, these tunes that really stick in your head, and then you try and interfere with those with kind of auditory, like dual task procedures. I've never been able to re-find that paper, so I have no idea whether I just dreamed this thing up or whether it actually happened and never got published. So I think potentially yes, but I don't think it's been done yet. But I think there's kind of then a more general question that comes out of that as to how far... Okay, so visual flashbacks, okay, maybe you can address that with visual stimuli, but then to what extent are flashbacks just visual, or are there other bodily sensations that are coming in there as well? And then do you need different approaches to try and deal with those? Or is the visual one the dominant one for humans because we're such visual creatures, and therefore that would deal with everything else? And I think those questions just haven't been answered yet. But really nice question, thank you. Oh, yeah.
Another question.
So ketamine and other psychedelics are showing promise for drug addiction, PTSD, and compulsive disorders. Do you think they're working through reconsolidation, and so how?
I would love to test that. So ketamine, I don't know so much, but certainly the psychedelics. One of the arguments for how psychedelics are working is kind of relaxed beliefs. I can't remember exactly what REBUS stands for, but the Robin Carhart Harris, Carl Friston kind of view, that what you're doing with psychedelics is relaxing the priors such that you would bring more sensory evidence. It would normally be assimilated, put into a different memory, bringing that into something that can be incorporated into the current memory. So I would absolutely love to test that hypothesis. You need a really nice way of kind of working out what those prior expectations were, but then could you basically take an ineffective destabilisation procedure, ineffective reactivation, actually make it effective through co-administration of something like psychedelics? So yeah, I would love to do that experiment. We haven't done it, obviously, but I think there is scope that may be a mechanism by which psychedelics are working. I don't think the evidence is solid there, and there are a lot of potential issues with some of the psychedelic studies, especially as many people are kind of paying to go on these retreats and do the experiment themselves. And if you've invested, $6,000 to go and fly to South America to go and do a retreat, you're probably going to find it life-changing, regardless of what it actually does. But I'm not diminishing this. I think there actually is a really sort of interesting potential biological mechanism there. I just think there are challenges to doing that research. So it's kind of, I would love to do that. We haven't done it yet, but very good question.
let's thank Amy again. Now, you probably know, if you come to the exam